We fight fire with fire by finding the right bacterial viruses that target, infect, and then kill the pathogenic bacteria without harming the commensal bacteria.
Today, in our expert interview series, I'm chatting with Amanda Burkardt and Dr. Mayukh from PHIOGEN. PHIOGEN is a new biotechnology company that is developing bacteriophage therapeutics with the aim of conducting a clinical trial in the near future. Today, they'll be answering questions from our community about bacteriophage therapy for UTI and how people can sign up for more information about the upcoming trial. Thanks for joining me to answer our community's questions. We're excited to hear more about your work.
Well, thank you for having us, Melissa. And we're really excited to talk to you and the rest of the community that you represent as we are actively developing solutions to solve this truly unmet medical need.
So let's jump straight into some basics. What is bacteriophage therapy?
So, bacteriophage therapy is essentially the use of bacterial viruses to target and kill bacteria that cause disease. And this is a relatively novel way of fighting bacterial infections that don't require antibiotics. So, throughout the course of medicine, there have been two ways that clinicians and the medical industry have really tried to solve bacterial infections. And the first is to kill the active infection with an antibiotic. This has proven to be relatively effective over the course of medicine. But the challenge with antibiotics is they're small molecules. And so bacteria can quickly and often do evolve to become resistant to those antibiotics. And so they're only good for a one-time fix and can further drive resistance. And then on the other hand, you have the prevention of bacterial infections altogether with vaccines, which unfortunately only a fraction of pathogens are viable vaccine targets. And so it leaves this gaping hole in medicine with patients who suffer from a vicious cycle of untreatable infections. And so we've really tried to find a new way to develop solutions for recurrent infections. And so I kind of try to say we fight fire with fire by finding the right bacterial viruses that target, infect, and then kill the pathogenic bacteria without harming the commensal bacteria, something that antibiotics can't do. So it really keeps the microbiome intact. But it's a really novel way of thinking about developing an antimicrobial that combines all of the best science from a biologic without any of the harsh effects of an antibiotic.
Yeah, and we got a couple of technical questions. The first one is, can you explain transduction and other mechanisms behind the way phages work?
Go ahead, Mayukh.
First of all, thank you for having us. Yeah, so the phages broadly, there are two different kinds of phages. One is lysogenic phages, another one is the lytic phages. So lysogenic phages, those are associated with the transduction phenomenon. However, the lytic phages, those are the super killer phages that we define, and those are less likely to be associated with the transduction. So that's why for the therapeutic purpose, we specifically select the lytic phages, which only kills the bacteria.
Okay, that's good to know. What previous studies have been done in humans, and what did they show about the effectiveness of phage therapy?
Yeah, it's a great question because phages have actually been around for, well, as long as bacteria have been around. So phages are naturally occurring. They're part of our microbiome. In fact, we are inhaling phage right now. There's phage inside of us. If you go outside and drop your pen on the ground and pick it back up, it'll be covered in phages. But that's not a bad thing. It's a good thing. You can kind of think of phages as an evolutionary checks and balances system with bacteria. And so the real key is finding the right phages that target the disease causing bacteria. That is the whole crux of really developing a high coverage, high performing drug candidate. And so phages aren't new per se. They've been around for a long time. The new part is trying to figure out how to find them and enhance their capabilities to allow them to work in a broad spectrum manner, yet paradoxically still be very targeted.
Mm-hmm.
So bacteriophages have been used as a therapeutic since the early 1900s. They were discovered around the same time as antibiotics, but antibiotics were easier to produce. And there was a world war that was happening that accelerated the development of antibiotics, where phages were still a question mark as to what they really were.
Mm-hmm.
So phages were kind of relegated to Eastern European medicine, where it was kind of Soviet era technology. And so the Western world didn't really pick up on it. And because for the time being, antibiotics worked so well. So there's been a lot of clinical use of phage, but it's mainly been in one-off patients and kind of in Eastern European medicine was how it was seen. There have been several clinical trials that have happened across the different decades. Unfortunately, because phage are not small molecule, they can't be treated in the same way. They are a biologic, and because of that, they require different manufacturing capabilities, and they require an intimate understanding of how these truly living viruses interact with their bacterial host. And we just have not had the technology to enable that to happen. And so there have been some pretty famous clinical trials with phage that
Mm-hmm.
haven't produced the best outcomes, not because the science didn't work, but maybe it was the wrong population that was target population of patients that was selected. Clinical trial design wasn't good. The phage product itself wasn't handled properly, and so the phages were inactive upon administration. But there have also been more recently some clinical trials in the United States that have shown good phage efficacy. Most notably, Armada and Biomax. They have both produced positive results from their top line readouts from the beginnings of their phase 2 clinical trials. And so there have been some successes in a clinical trial, a more controlled stance, but overwhelmingly, phage have been used as kind of a last line as therapy in the United States, anyways.
Mm-hmm.
And that's because there is no FDA approved product yet. People are working on it. And so, because of that, it's been used in these emergency use cases. So, you can envision a patient, for example, that has a recurring UTI. They've had this UTI for years. They've taken all types of antibiotics. It won't go away. It's a debilitating infection, as many of the people in your community know.
Mm-hmm.
And so their clinician can actually ask academic labs, and some industry companies have the ability to do this as well, but it's normally been academic labs to find phages that target their bacterial strain, put together a cocktail, ask the FDA if they will grant an emergency use authorization. The FDA has 24 hours to respond. Phage has about 99% approval rating. And that patient can go forward with phage treatment that normally has a very high success rate. And so over the course of time, it's really been these emergency use cases that has built the case study. So it's a mix of these emergency use cases and now some controlled clinical trials that are coming to the forefront of how phage has been used and is being used.
Yeah, it's great to hear that there's been that progress. You mentioned that it can be part of the process is just finding the phages. Does each phage, is it specific to one bacterium or can it target multiple strains?
So there are a couple of phages that can target multiple species of the bacteria. However, most of the phages, those are species specific. And within that species, also you can select the phage which can target most of the strains which are moving around the globe. So we have been selected the phages in a specific way, and since we are making a fixed product, so that can have a maximum coverage throughout the globe.
Okay, that's good to know. And do you know if phages have been developed for specific organisms? So some of the common ones for UTI, Pseudomonas or Klebsiella?
Yeah, I mean, so there are phages for the E. coli, there are phages for the Klebsiella, phages for the Pseudomonas. Also, these are the gram negatives. And also for in the gram positive, the Staphylococcus aureus, there are phages for that as well. Yeah.
And could phage therapy technically be used in any location in the body?
Um, yeah, that is true. I mean, it can be used for any locations. However, the administration of the phage that needs to be specific and appropriate, I would say, for a location. As for example, for the respiratory, maybe the nebulizer. So that would be the route of administration, or maybe the in the bladder where you have the bladder infection, maybe the bladder instillation. That could be the appropriate way, because the reason is wherever you have the bacteria, the phage needs to go there. That's how the phage works. Phage needs to see, it's a numbers game. Phage needs to see the bacteria to produce its maximum efficacy. So if they see each other, then only phage will attack the bacteria, and then only they will kill the bacteria, and they will produce more phage to kill more bacteria. So they need to see each other.
Okay. Does there need to be a certain minimum level of bacteria for the phages to act in that way?
That's true. That's true.
How would that work for someone who has a chronic infection and maybe low levels of bacteria chronically over time?
So let's say if that infection, like chronic infection, is if it is a systemic, which is a bacteremia infection, so you can use the IV for the bacteremia, which will directly go to the blood. And if that person has a minimum level of bacterial infection there, you can use more phages to reach out throughout the system, which wherever you can find the bacteria, that phage can kill the bacteria.
Okay. And you mentioned that once the phages are there, if there's enough bacteria, the phages will multiply to continue doing that work. Is there a chance that they might attack the healthy microbiome in the body?
Um, so that chance is very slim. I mean, and more specifically, the way PHIOGEN, we select the phage which only kills the pathogenic bacteria. That phage does not kill the commensal bacteria, which is your microbiome.
Okay, so once the organism that the phage is targeting is eradicated, what happens to the phage then? Do they remain in the body?
Um, they have a certain time frame, the certain time that they remain in the body. After that, they actually release the phages.
Okay. Is it possible that phages could move from one site in the body to another?
That's true. I mean, there are different phages. They have a more, depending on their size, they have the more transferability, but there are some phages which can release through the excretory system very easily.
Okay. And has it been shown that phages can attack bacteria that are within a biofilm?
Yes. So that's a very good question, actually. So some of the disease indication, those are associated with the biofilm, like our UTI also. So we have a specific phage which has the anti-biofilm capability. So it can break the biofilm, and then it can go inside and attack the bacteria.
How did you discover that these phages could actually attack organisms within a biofilm?
Yeah, that's a great question. Actually, we are the platform-based technology. We have 6 text platform, and one of the step is a bioclimatic screening, where we create human-like environment in vitro, which is basically in the laboratory, and then we use our phages in that specific environment and find out how phages work in that specific environment. That's how we select the phages. So we develop the biofilm in the lab on the plate, and then we use the phages. We use multiple phages, and then the best performer, we select that. So that's how one of the phage that we have in our cocktail, in our drug, we selected that phages. When we use that phage for the regular assay, that phage performed really, really poor, really bad. So we did not select that. But when we use the same phage in that specific environment in the urine where you had the biofilm, that phage worked really well. So that's how we selected that, and that's the beauty of our bioclimatic screening.
Yeah, I think that's really important for the people in our community because a lot of them probably do have biofilms with their infections.
Yes.
But that also brings me to eligibility for phage therapy. Is there something that makes one person a better candidate for it than someone else?
It's a good question. Overwhelmingly, the reason why people turn to phage therapy is because standard of care has failed. So that means that antibiotics aren't working on that person's infection, or that person is chronically colonized. And so they're never able to get rid of the infection because they self-inoculate. So, for example, UTIs. Overwhelmingly, a majority of UTIs are from self-inoculation. The E. coli causing the disease is coming from that person's GI tract, or there's a reservoir, possibly in the vaginal cavity, or they are embedded in the cells of the bladder, or that person has a drug-resistant bug, or the infection is never clearing to begin with because of biofilms, or because the bacteria are embedded in deep layers of the bladder cells. And so there's a lot of different reasons why people may or may not respond to standard of care treatment like antibiotics. And so that is really why we're focusing on recurrent UTIs, because for the most part, people who, specifically women, who get uncomplicated UTIs can take an antibiotic, clear it, and you won't have another problem. But there is about 40% of that population who, after they take that first antibiotic, they get another infection and then another infection. And then before they know it, they're chronically fighting these recurrent infections. And so we've strategically really focused on recurrent UTI because there is, by definition, an unmet medical need. There is no treatment options for these patients that are suffering, and it's debilitating. I've suffered from recurrent UTIs in the past, and luckily I found a treatment option that worked for me. But I just got a small snippet of the true hell that some of these people live through every day, and I couldn't imagine trying to live a functional life going through this every other week, every other month, even every 6 months. It's a huge burden financially, and productivity wise, as well as welfare. So the patients that are best suited for phage therapy, in my opinion, are the ones that suffer from recurrent infections where, for whatever reason, standard of care has failed. At the moment, phage is really seen as a last line option for a healthcare solution. I really hope that phage is brought as a first line solution. And so that way we're able to really steward antibiotics in a better way and really hold them for the infections that don't respond to phage therapy. And there's literature to suggest that that could be a viable option someday later down the line. But phage just has to become more mainstream first before that can happen.
Yeah, it's an interesting point because a lot of people in this community who have chronic infections, they often culture negative. Do you need to be able to culture to be able to develop a phage therapy treatment, or is there like an off-the-shelf option that might work for those people?
So the off-the-shelf product option, so the way phage works, as Amanda mentioned, it is very different than the small molecule. So that's why even the standard of care, if we bring the phage as a first line of treatment, the standard of care needs to be modified according to the way phage works. So that's the way we select the drug. Since we are the platform-based company, we select the disease first. As for example, here UTI, and we specifically trying to target the recurrent UTI. And then we're trying to find out what are the complications associated with that disease. And then we select the bacteriophage, which has the ability to target each of the complications. As for example, for the UTI, so biofilm is one of the complications. Then the recurrent UTI, the recurrent event of the infections. And that can happen because of many reasons. As Abinda mentions, that sometimes, reinfection, and also sometimes you have low bacterial infections which is there inside the tissues. The antibiotic doesn't have ability to reach that, so that can reinfect and the patient. So that's how we select each of the phages, and we put together and make our drug. So if you go with that kind of mechanism, so you will see the maximum efficacy for your drug product. So that's why we are confident that our of the shelf drug product will show maximum efficacy against this specific disease indication.
Does that mean in the future it might be possible for even someone who does culture negative but has symptoms of a UTI to try phage therapy because it's an off-the-shelf product and it doesn't need to be matched specifically to the organisms found in their urine?
Well, I mean, the way we are trying to target our coverage in terms of host range is almost 90%. So, and that is a global. So since we are trying to make the global product, we are trying to cover as much as strains possible which are pathogenic and distributed globally.
Yeah, great. It sounds really promising. One of the concerns people often raise in our community is that they don't culture positive. So they never have had a positive culture, and they're concerned that means they won't be able to access phage therapy. Do you think that's the case in the long run for them, or will they be able to access it in the future?
It's an important point because overwhelmingly, the data suggests that most UTIs are caused by E. coli. However, with the work that you've done, Melissa, with your patient community, I think there's learnings and discoveries that E. coli is biased for being picked up on diagnostics. And maybe there are other bugs that have a higher percentage, and that actually maybe E. coli isn't as widespread as previously thought. And so, although now data suggests that that is the case, anywhere from about 85 to 90% of all UTIs at the moment are diagnosed from predominantly E. coli. This is really the target of our product that we're developing, is really to focus on recurrent infections from E. coli, mainly because we're a small company, we have limited resources. And in order for phage therapy to get to the next round, we have to have small wins in order to get investor confidence, patient buy-in, and clinician support. And so we have somewhat narrowly defined our field goals that we can make sure that we hit. So for those patients that maybe don't get a positive culture test, the end goal is to someday for us be able to create a product that doesn't just hit E. coli, but hits the other bugs that also cause recurrent infections that some people may never get a positive culture from. And so although our initial target patient population is really recurrent UTIs with a focus on E. coli, it is in our pipeline, and we actively have researchers that are working on developing products that can hit those infections that are polymicrobial, that maybe E. coli isn't the main bug. It's a little later down the road for us, but we recognize that that is a group of patients that also need access to this type of recurrent UTI solutions.
Definitely. Well, let's talk a little bit about how phage therapy is administered. Mayukh, you mentioned earlier that you might, if it was respiratory, you might take it in a certain way. Is it possible that you could take an oral phage therapy drug for a bladder infection, or do you think it needs to be via instillation?
Yeah, it is possible. We are still figuring out since we are in the process to develop the clinical development plan for our clinical trial. That's why we want to have this survey. We want to gather that patient's perspective and how they prefer to receive the phage therapy. So based on that, we can do the experiments in the lab and find out what is the best way to go, and what's the best way we can offer the treatment to the patient.
Yeah, I guess it's a good time to talk about the study that's currently running as preliminary research for the clinical trial. Do you want to provide some insights on that?
For the clinical trial, I can say that we are still working on to develop the clinical trial with our clinical advisors. We plan to start the clinical trial Q4 next year. I would say that more to come and stay tuned.
Mhm. Yeah. And really, we're thankful, Melissa, that you're so willing to open up your network and your community to inform us. One of the things that we as a team, Mayukh and I identified early on, is a lot of these companies that are developing solutions for patients, no matter the indication, don't talk to the patient first.
Mm.
And that shows when it comes time for them to administer their drug product, or even when it comes to FDA approval and then launch, if the patient isn't kept at the center of the discussion, can really run into problems later down the road. And so we identified that early on and made it a point to seek out the best of the best, which of course is you, Melissa, and the beautiful community that you've built. And most importantly, it's science focused, which is something that you don't find often. So kudos for really building something that is bringing true scientific evidence and discussions to patients that were pretty much relegated to searching Facebook groups or other avenues of just trying to find relief. And so this survey that we will be conducting, anyone who takes it, which I hope everyone who's interested does take it, will be single-handedly informing how we develop our clinical trial and how we develop our drug product to meet the patient's needs. And I think that's really important and really speaks to the community that you've built. So thank you for allowing us to be able to work with your network that way.
Yeah, I think it's really good that you're doing this preliminary research so people who are interested in the trial that will hopefully run next year can actually provide their opinions and share more about their experiences. So anyone who is watching this that would like to participate in that study, we'll make sure we put the link to that in the video description below, so they can join in and then also sign up to get more information about the clinical trial as you progress towards that. And so I think it's the best approach for this because the patients in the community are really the ones who need this and therefore can tell you how they think it should be run in the way that will be of most benefit to them. So I'm also very glad to see that that's the approach you're taking. It's also the reason that we're doing this interview, because the announcement of that study resulted in so many questions about phage therapy. And so we do have some more about how phage therapy works. And one of those was how long it usually takes to treat a UTI with phage therapy.
Yeah, at the current moment, we are still trying to identify what the best dosing and treatment course is. At the moment, we're working with our clinical advisors to figure out what that looks like. We don't imagine it would be more than a 3-day dosing, but I would hate to put a time limit on it just yet until we actually know from a clinical evidence-based decision making of really what the best timeline would be, what the best dosing regimen is. Our main goal is to make it as non-invasive, as less painful, and as much compliance as we can, and to make sure that the clinicians are happy with it too, and that they're happy to have patients come into their clinic and administer it in a way that makes both the patient and clinician confident in that administration protocol. So we're really leaning on them. And these clinicians are actively treating patients. So they're infectious disease docs, they're urologists, they're urogynecologists, these folks that we're reaching out to and leaning on for their expertise to make sure that we have the clinician as part of this discussion as well is really important to us too.
Yeah, that makes sense. Do you know at this stage whether external factors like diet or urine pH affect the success of phage treatment?
At this moment, so we are still discussing with our clinical advisors and putting that in the exclusion-inclusion criteria for our clinical trial. However, we do not identify those as a potential problem, but we haven't finalized yet.
And has there been any research previously that have shown any side effects from phage therapy?
Yeah, that's a question that we get a lot, especially from those who are interested in it from a patient standpoint. You can look at a lot of the available literature and even review many of the clinical trials that have happened. And overwhelmingly, phage is regarded as safe. Um, there have been little to no serious adverse events that have been documented in a lot of these clinical trials, but also in the EIND cases uh, for a few different reasons. As I mentioned earlier, phage are naturally occurring. So we're always exposed to phage. And because of that, it wouldn't make sense to have a strong inflammatory response, or else we would always be inflamed.
Mm-hmm.
They're part of our microbiome. Um, of course, we go over and beyond to ensure that the phages that we are using are safe. And it's actually part of our selection criteria that the phages that we select don't elicit neutralizing antibodies and a few of these other factors to make sure that we are selecting the highest performing, highest quality phages. But overwhelmingly, phage are generally regarded as safe. And from all intents and purposes, I'm pretty much widely known, phage are relatively seen to not really have serious adverse events that are documented. If there are, they are relatively benign, I would say. Possibly a fever might be
Mm-hmm.
indicated or inflammation around the site of infection. But besides that, I don't know of any really serious documented events.
And how does the immune system interact with phages?
Usually, the immune system, as Amanda mentioned, that there are some evidences that specific phages they have the neutralizing effect, which um, produced by the human immune system. So, the platform technology that we have, we select the specific phages which doesn't have the neutralizing effect from the human
Mm-hmm.
body. So that's why wherever we see the neutralizing effect, we uh, exclude that specific phages.
Okay.
So considering that, we do not expect any neutralizing immune response from the patient.
Okay.
Um, yeah,
Well, that's interesting that the immune system reacts to some phages and not others in that way.
right, exactly. Yeah.
Has there been any research into or any comparative studies into whether off-the-shelf phages perform as well as customized phages?
That's a very good question, actually. To my knowledge, I don't think that there is a specific study have been performed just to compare like a personalized versus the off the shelf. Yeah,
Mm-hmm.
I really don't think so.
Yeah, it doesn't seem like a lot of research has been done in this space in those kinds of terms, so it's not surprising to hear that. But have you done research with your own products in humans already?
Yeah, well, I mean, the part of the product that have already been used through our academic arm, which is the Taylor Lab,
Mm-hmm.
to treat different compassionate cases through emergency use through FDA. And we have treated multiple patients. So far, they have treated, I think, more than 30 patients,
Mm-hmm.
and some of them are with the specific disease indication that we are targeting, which is the recurrent UTI.
Okay,
And we have seen a good success over there.
great.
So that result is very encouraging, actually. So that's why we are
Yeah, it's really good to
confident
hear.
that our product will have a good success in the clinic.
Yeah, it's really great to hear. And so your clinical trial that will hopefully run next year, where are you thinking those sites might be? Will it be US specific or will there be global sites as well?
Yeah, we have intentionally built our product really to focus on as widespread coverage as possible. We recognize that recurrent UTIs don't only happen in the United States. It's a global phenomenon. UTIs are one of the most common bacterial infections worldwide, with over 400 million cases every year. And so, intentionally, we are actively building our product to have a global reach. Majority of the strains that we have are from the United States because that's where we're headquartered. But we really believe in building a product that doesn't just have a solution for high income countries. We are actively screening our lead product against low and middle income countries. So E. coli strains that were collected from UTIs all over the world, and really developing and optimizing our drug candidate to have as global and far spread a reach as possible.
That's really good to hear. Where the clinical trial sites are, will patients or candidates have to travel to the site for administration of the phage therapy?
At the current moment, that's looking like that will have to be part of our trial design. We're still sussing out route of administration, but there will probably need to be, we're assuming it will be outpatient. So patient goes to the clinic, receives the phage therapy, and then leaves. But it would have to be done in some type of clinical setting. And so there would probably be travel that's required to wherever the clinical trial sites are.
Okay, that's helpful. And do you think there's a possibility that phage therapy could not only treat infection, but also prevent future infections?
It's a really important question, and actually the crux of what we're doing as a company. As Mayukh had mentioned earlier, the key piece to developing a solution is not a one-time fix. Antibiotics had tried that, and unfortunately, bacteria are smarter, and they found a way to undermine antibiotics. And so, really, you get this patient population that has these recurrent infections. And so, it's really important that if you are developing a solution, you can't be as good as standard of care. You are held to a higher standard. You must be better. And for these recurrent infections, that's what the treatment solution demands. And so we intentionally going into this knew that if we're going to build a product, a true drug candidate for this target indication, that we would have to build something that treated and prevented, and did it in a way that no other company has done yet. We're still in the process of really discovering what we have been able to do from a scientific standpoint. But we believe if we're successful with this clinical trial, that we can prove not only does our product kill the active infection, but it simultaneously primes your immune system to form an immune response against the infecting bacterium. So that's a lot of jargon, but I always say if an antibiotic and a vaccine had a baby, that's essentially what we've created. And so we've meticulously built what we believe is a first in class as an immunizing therapeutic. And the nuances to that we're still disclosing and trying to protect from an intellectual property perspective. But to your question, it is paramount that you treat and prevent recurrent infections, especially for those suffering from recurrent infections, because they don't just want a one-time fix. Because the infection will come back. And so if you're able to treat and possibly immunize against that infecting strain, that can completely change the paradigm of how we treat not just recurrent infections, but recurrent UTI infection, recurrent urinary tract infections, but then potentially all bacterial infections moving forward. But we want to try to hit our mark first. And so we're starting with recurrent UTI.
And we're glad that you're starting with that. And I think the prevention piece is really important for this community, like you said. And you mentioned earlier that patients have been able to access your products through certain clinicians and colleagues. In the meantime, before the trial launches, is it possible for people from our community to access the products?
Yeah, it's something that we're working on with our partner lab at the moment, which you've had on this interview before, the Dr. Anthony Moresso's group, Taylor Lab. They have utilized this emergency use authorization to treat patients in the past. They will be, excitingly, moving to a brand new facility where they will be able to manufacture GMP grade phage. So in the meantime, within the next 2 to 3 months, they will be busy packing up their things and moving down the street to that brand new facility. And so I would normally say that any patients who are interested in accessing some of these phages could reach out to Taylor. They still can, but the likelihood of them being able to manufacture will probably be low because they're moving to a new facility that will increase their manufacturing capacity to be able to do that more accessibly. So for now, probably not before the clinical trial in any meaningful manner do I think patients would be able to access these phages. But please rest assured knowing that every day Mayukh and I go to bed and wake up, we're actively working to get this money raised to fund this clinical trial. So anyone listening to this that has the ability to access our clinical trial, that we will actively be working to make it available just as soon as possible.
Yeah, that's really good to hear. I think this is a good point to leave the interview, but just to again mention the study that's currently running that will help you design the clinical trial. So anyone that is interested in contributing to that and potentially participating in the trial, we really encourage you to fill out the survey, and you can find the link in the video description below. And I wanted to thank you both again for joining me to answer all the questions we received.
Thank you for having us, Melissa.
Thank you for having us.
Thanks so much for watching. I hope you found this expert video helpful. If you'd like to learn more about this or related topics, be sure to check out our other videos or head over to liveUTIfree.com for related articles. We'll drop some links in the video description. If you like what we're doing on this channel, you can support our work by hitting subscribe here on YouTube, and don't forget to tick the bell so you'll be notified of our future videos. Thanks again for watching, and until next time, keep asking questions and pushing for better solutions.